So as I think that we're on program, let's start. Dear artists, dear researchers, dear both, dear all, in my capacity as a representative of the host here, the University of Applied Arts, Vienna, and the ZENTRUM FOKUS FORSCHUNG, I'm pleased to open today's exhibition. An investigative exhibition that once again opens our senses and thus opens up paths to understand differently, to sharpen our perception, to understand not as we used to, and thus to allow us to recognize the other. This exhibition is the result of an immensely committed research project at the Institute of Architecture of the University of Applied Arts, supported by the Austrian Science Fund through the renowned program for artistic research, the PEAK program, which is unparalleled internationally. Please allow me this side note. I believe it is a necessity to show how artistic practice transforms understanding and thereby also research. And it is always amazing how this is dominantly also neglected, to ignore, or in other words, how to consult art and its explorative capacity allows also when conducting research seems to me is a pity to ignore this fact. Unfortunately, this is still a widely established practice, but luckily not by the Austrian Science Fund, the FWF, not in the practice of research such as it is happening at Angewandte, and certainly not in the work of the project's leading team, Barbara Imhoff, Daniela Mitterberger, and Tiziano Derme. Artistic and architectural practice is in the center of the focus of this research here, to explore as it needs to be, as it is required. It is thanks to these three, Barbara, Daniela, Tiziano, that we are standing here today after years of research and development, and of course also thanks to the partners who are also here today and who can best present themselves, their positions and their views of, to and on the project. A big thank you to all of you. So now we may experience how nonverbal communication between humans and bacteria, the perception of shared space, sensing and coexistence of different sensory modes and reactions were explored. Of, for example, A, cyanobacteria, and B, Ecoli bacteria, and an eye tracking interface, which brings us to the attention of the attended. Just to mention maybe one aspect, I'm curious. Therefore, let me conclude and thank the ZFF team for their work, especially Mariana Mondelos and also Felipe Duque and all involved colleagues of the EOA at Angewandte that this was made possible, and with heartfelt gratitude to Daniela Mitterberger and Tiziano Derme, I still remember with joy our first encounters, and Barbara Imhoff with all her experience in expanding the possible and pointing our attention to untrodden paths. Thank you for this. So I hand over to you again to be guided by your ideas of exploration to add some further degrees of life. Barbara, please.
Thank you, Alexander Damjanic, who is the head of ZENTRUM FOKUS FORSCHUNG at the University of Applied Arts. We are here in a small pavilion in the Rustenschacher Allee, and this was originally the studio of the sculptor Fritz Wottruber. And here we're showing the exhibition Degrees of Life, which is part of Co-Corporeality. Co-Corporeality is FWF Austrian Research Fund funded project, and it is sort of at the intersection of art, architecture, and natural sciences. And what you will see here, we will walk with you through the exhibition, and you will see all the players and the actors of the team of the people who made it possible. The research focuses on communication with our microbial environment inside of us and outside. And we are trying to sort of go a little bit beyond and sensitize ourselves to start this kind of communication, but also to feel the presence of the two bodies. So we are trying to sort of get into the living room of these microbial environments, of the the microbes. And we also will try to make a sort of jump in scale, in a time scale, which is a little bit more than just a time zone jump. And so we're sending a sort of, we're not really sending a diplomatic delegation to these microbes because we cannot, but we will use other means through tracking and sensor systems to start the communication. And yeah, with this, let's take a look and see how we can communicate with sort of species which are a little bit more alien to us than we think.
Thank you, Barbara. Hello, I'm Daniela Mitterberger. I'm part of the Co-Corporeality leading team. I'm part of the Angewandte, and I'm happy to show you our exhibition, Degrees of Life. And it all started with a big question, how we can create an architecture that can react to us, react to inhabitants. And then we discovered if we want to really do that, then actually we need to discover what creates biomaterials, what is out there we can communicate with. And actually we discovered that we have to start to learn how to communicate with bacteria. And we found different kind of bacteria here that we can communicate with, and different kind of questions on how we can visualize this. So not just how we can communicate, but also how we can see that. Bacteria are incredibly small, so it's very difficult for our primary sense, the eye, to understand what's going on. So we have different topics at different locations in this exhibition that show in a big scale how this could look like. So we have Ecoli here, we have Bacterial Cellulose, cyanobacteria. So all different kinds of bacteria that require different kind of input and a different kind of communication medium. And I'm gonna directly lead to the first type of bacteria, which are the cyanobacteria, and our expert Judith, which will explain all about it and all the world that lies behind the research that we're doing with the cyanobacteria.
Thank you, Daniela. I'm Judith Asche-Jenon, microbiologist at the University of Innsbruck. And for this special project, we dived into the fascinating world of cyanobacteria, known also as green-blue algae. And they're the architects of higher life forms since they started to produce oxygen. And that's why 3.8 billion years ago, they contributed to form the Earth's atmosphere. And they're able to produce oxygen by the complex mechanisms of photosynthesis. And this complex process is actually driven by light supply and by the light quality. And the different quality of light in terms of light intensity and photosynthetic active radiation is triggering the development of different pigments. And cyanobacteria possess 3 types of pigments. The green one, chlorophyll A, the red one, phycoerythrin, and the blue one, phycocyanin. And this was basically the concept of our project. And here we see these different shades of green. Now I pass to Tiziana. Thank you.
Hi, thank you, Judith. What really was driving us to run this project fundamentally was a curiosity on how do we understand this idea of co-corporealism, no? An idea of where 2 biological subjects are looking at each other, naming us as humans versus the microbial world. And the way how we thought this exhibition was to articulate and to scatter these environments that are understood as closed environments that are able to somehow protect and satisfy the requirements that the bacteria, this microbial world, has. No? So what we see here is a juxtaposition between different kind of presence. The biological presence, which are depicted by the presence of different bacteria and us as visitors, and of course the machining presence. Machines in this case are the mediating factor between us and the microbial world. And it's interesting that this mediation doesn't happen only through the machine, but also through our body. No, so the visitors are actually the actors and the agents to directly interact.
Thank you, Tiziano. My name is Heribert Insam. I'm from the University of Innsbruck, and I'm appalled by blue-green algae, by cyanobacteria that have started to form life on Earth. And if you look at this bacterial mat, this cyanobacterial mat, you can imagine that many billion years ago, they started to make our planet hospitable. They started to form oxygen. However, they were used to having light once a day. They had 12 hours of sunlight, 12 hours of night. And now we are changing the perception of these cyanobacteria. They are now experiencing our presence, and our presence has the effect that there is a weird photoperiod. The photoperiod changes within very short times, and we have installed sensors in there. And in this 60-liter tank, there is this very small sensor on one of the edges, one for oxygen and one for the pH. And if the camera is going to look at the screen in the back, you can see how this photoperiod affects the production of oxygen and the pH change that occurs within seconds or within a few seconds after the habitat is changed by stopping the light. Photosynthesis is stopped at once, and photosynthesis is the process that has enabled life on Earth. So with this ancient organism, we are able to communicate today. And now I hand over to a bacterial world.
Hello, my name is David Berry, professor at the Center for Microbiology and Environmental System Science at the University of Vienna. Each of us is home to a rich and vast microbial ecosystem within us, the gut microbiome. And the gut microbiome is essential for health and provides us nutrients. And we in turn provide nutrients to the microbiome. And so this is a true co-corporeality in its most physical sense. And here we display this, showing E. coli in each of these containers. And we have grown these E. coli with a sugar radium, which it enjoys very much. And it produces acetate from these sugars. And this acetate is an important nutrient source for us that we would take up in our bodies. Unfortunately, in this process, E. coli acidifies the medium and destroys its own environment. And in homeostasis in our bodies, we would re-equilibrate the system. And here we can see using pH-sensitive dyes, when the medium turns from purple to yellow, E. coli has acidified the medium, is unable to perform its growth. And here using eye tracking software system, we're able to identify which of these containers have now been acidified and move then an automated dispensing system to add a base and re-equilibrate the system and allow E. coli to continue to grow. I will now hand over to Damien.
Hi, I'm Daniel Milowski, part of the core team at the Angewandte, and I was in charge of mostly designing and building the machines that are making the interface between the human and the different bacteria and the biological world, so to speak. And basically, they are created from adaptation from 3D printers that then got scaled in size and equipped with different elements like syringes or spraying mechanisms that then again get controlled or triggered by behaviors that are controlled from a centralized position based on the human gaze and interaction with the machines. Daniela.
Thank you, Damian. So I'm already wearing it. So we talked before about the non-verbal communication, but that we're actually using the gaze. And I'm wearing a custom eye tracking device that we're setting up. And if you can follow me here, you actually see what it's doing. So we have 3 different cameras, one towards the eye, one towards the room, like 2 towards the room. And here is actually where we do the calibration. Here you can see my eye and actually also the position in real time. And what it can detect is the position of the eye, the direction of the look, but also unconscious data such as, for instance, the pupil diameter. And we are using the look, so the gaze, to actually establish the interaction with the machines. And all machines react differently. And you can also see on the interface here, you can see my perspective of the room and also the moment when I look at machines. So different machines do different things. So here we can control the light. Here we can control which syringe is depositing chemicals, and here the nutrient deposition. And I will then give the word to Martin, who will talk more about the sensor system, his work, also on the eye tracking.
Hi, my name is Martin Gasser. I'm a software developer and researcher, and I'm associated with the Austrian Research Institute for Artificial Intelligence and also the Angewandte. In Co-Corporeality, we have, as has been already mentioned, tried to build interfaces between humans and microbial life forms. So when you try to do that, you're actually trying to establish communication between alien life forms. So you need some kind of translator. And we try to use actually 2 modes of translation, right? The first mode of interaction or translation was based on emotion or facial expression. And so we built this device, it's called the Efeeder, and it's a robotic system that can nurture microbial life forms based on information gathered from human faces. So if you look happy, if you look nice, the life form gets a drop of a nurturing solution. If you feel sad or angry, it gets a shot of UV light. So this is a very simple and crude form of interaction, but I think you get the idea. And the other form of interaction was, as has been already mentioned, spatial interaction in space. And in order to do that, we built an eye tracking system that can first detect your position in space and the orientation of your head. And second, the orientation of viewing direction of your eye. And here we can see a video that demonstrates how the system works. So this is an early prototype we built last year. For this exhibition, we have built an advanced version of it. So it's based on, we have used a bike helmet and mounted 3 cameras. So it's 1 camera pointing to the eye of the person wearing it, and 2 cameras pointing into the world direction. And one of the cameras is used for detecting the orientation and the position of the viewer, and the other camera is used for calibration of the system. So it's a regular RGB camera. And with this device, you can actually detect the position of a person wearing it in space and where the person is looking at. And based on this information, we tried to establish a way of non-verbal communication between humans and microbes. Now we'll hand over to Neptune.
Martin. Hi, I'm Neptune Yosefi. I'm a PhD student at the Materials Science Institute at the University of Vienna. During this project, I was mainly working on bacterial cellulose, but first I'm going to tell you a bit of the Efeeder, how it works. So the Efeeder triggered 3 reactions. So the bacteria are exposed to lactose, X-gal, or UV light. So what lactose does, it's basically sugar. Bacteria eat the sugar, and once it's eaten, they release acid, and the acid changes the color into a red color. X-gal, which is also a sugar, but it's attached to a pigment. Once the sugar is eaten by the bacteria, the pigment is released and it changes into a blue color. And the last thing is the UV light, which when exposed to the bacteria, they die. All right. So, but I was mainly working on the bacterial cellulose side. So usually bacterial cellulose will go on the interface, as you can see here. And also later you will see that, but we were trying to make them grow on objects. So what we did was first 3D printing an object, then coating them with dopamine, and through shaking, we would end up having bacterial cellulose on the objects. This bacteria, however, is in a very early stage, so this will happen hopefully later. And now we're going to continue with Tiziano. And he will talk about the bacterial cellulose a bit more.
Thank you, Neptune. So as Neptune mentioned already, here with the bacterial cellulose, we are really unpacking a different level of the project, a level that looks at the intersection between architecture and new material systems. Yeah. So bacterial cellulose is a widely renowned biopolymer that is currently investigated from different institutes as alternative to, for example, plastics. So here what we see are different closed environments that are used to grow this material, literally. So it's a material that is produced through the metabolism of very specific bacteria. Yeah? And the aim of these enclosed environments is also, of course, host the capacity to sustain themselves. No? So the machine in this case is an aerosol system that is providing the nutrients continuously, allowing the material to grow. Yeah. While, for example, in this other case, of course, the investigation is oriented towards more towards a design aspect. No? So how can we integrate different technologies and enhance new design perspectives with this material? So I hand over the word to Javier. Yeah.
Hi, I'm Zaid Madden. I'm a Master's of Architecture student at the University of Applied Arts, Vienna. And towards the end of this project, I was working on the lattice structure that you see here for bacterial cellulose, where we were trying to create a structure which not only supported the bacterial cellulose, but would adapt with its changes. So this meant that we needed to create a flexible structure, which led us down a path of creating a custom structure. So this meant that we used a method, a specific 3D printing method, which allowed us to print a flexible material, a modular system, which you see laid out here. And the idea is that this structure will enable this bacterial cellulose to grow around it and above it. And if you remember the prototype from before, when it dries out, it will kind of deform and change shape. Now I'll pass on.
Hello, I'm Marianne Munders, working at ZENTRUM FOKUS FORSCHUNG and also taking care of the chat now from the screen. So if you have any questions, feel free to post them now and we will answer them. No questions yet, but feel free to write also comment, statement, whatever you feel like in the chat.
Oh, there's one question, one moment.
They ask, hello, I'm curious about the bacterial cellulose and the process behind its production. Whom should I pass on?
First to Tiziano.
Tiziano. Someone have a question?
Yeah.
What's the question?
For the cellulose.
Okay.
How to produce?
How to produce it, okay. So we use two different types of cellulose, bacterial cellulose actually. Depending on which one, I would definitely recommend to use the kombucha because that is super easy. So you buy the kombucha tea from the pharmacy. And the medium is basically sugar, there's the water, vinegar, and we used green tea to make it very white. But usually you can use a mixture of green tea and black tea, and that's it. They need room temperature, and yeah, it should be sterile ideally, because otherwise other pigs will go there, since the medium is very rich in nutrients. That answers your question, so I missed it. Ivan is asking what kind of bacteria is that? The kombucha is an Acinetobacter bacteria, and the other strain we used is called Sucrofermentus. So these are bacterial cellulose producing bacteria. So, but you can read the book if you're interested. Thanks. One more question by Leo One. How will the project develop further?
Yeah, I think I just wanted to, so the project is being developed further and will also conclude in a book. So there's much more information on it. It's same, the project title Co-Corporeality on humans, machines, and microbes. And I think the question, how can the project be developed further? Do you want to say something about that, Daniela or Tiziano?
Sure. I think more experiments, much bigger experiments. I think one of the biggest advantages of the Co-Corporeality project was to jump in scales. And I think to have experiments in architecture scale is super important, especially when it comes to bacteria, because in that moment you actually create new questions that were not there before when it was in the Petri dish. So definitely jumping in scale and also jumping in time periods to actually have experiments run for a longer time period. And this allows you to completely establish new experiments with these bacteria. Jana, you want to add something?
Yeah, I mean, like Daniela mentioned, for sure, the issue of scale is something that we are really interested in, of course, because by weaving research from life science, material science, machine learning, and architecture, there is also a natural projection to a more architectural scale. So of course, the idea of scale, and of course, I would suggest also the idea of interaction. No, So what is the role as a humans we have within an architecture that can perhaps respond, learn, grow, and those are all are questions that we find very architectural. Thank you.
First question.
Yeah, we have one more question by Edward Curry. What is the timing typically to grow these bacterial labs? Thank you.
Excellent question. So the timing varies very much depending on which bacteria you're talking about. So bacteria like Ecoli that are living in our guts typically divide very quickly. That's on the order of every 20 minutes, 20 to 30 minutes. So we can grow a culture like this overnight in optimal conditions. Environmental microbes, however, typically grow on much longer time scales. So these displays that you see here are grown on the order of weeks. So this is very dependent on the type of microbe you're talking about.
So are there any further questions? Last chance to write them down. Maybe for seconds. There's a follow-up question by Ivan, if you would write them at the end. But you don't try it at the end or with the exact wording.
To get them, yeah, this question is actually. Would you like to address the drawing of the bacterial cellulose? The drawing of the bacterial cellulose? Yeah. They're asking if you're going to try the.
Maybe you can show the thing where you draw.
Yeah, so basically, I mean, what you see here are basically environments of growing for the materials, but of course there is a part that we don't see yet, which is what we're going to do with the material afterwards. And normally the bacterial cellulose is a biopolymer that can be used, as a natural membrane. No? So what we do normally is to remove the microbial mat from the culture, neutralize the culture, and let it dry at room temperature. Yeah, this is favoring the evaporation of water, and of course is strengthening the supplementary structure, like we see here. Of course, as approach this, we find as direct implications into how do we use natural materials in architecture.
Any more questions? No, then I would go to Barbara again. Oh, no, it's just one came in, let's check. By Helga Eichmeier, maybe you have already given the information. How long did the setup of the exhibition grow? Like all of the different bacteria, how long do a dialogue situation need to grow so that an interaction is visible?
Well, I can speak for the Ecoli experiment here. So we began this morning at 7 in the morning, and at that time, all of these containers were dark purple color, and we came here about 10 hours later. Already, the ones with the most Ecoli had turned yellow, And what we expect to see then is over the night that the ones with fewer E. coli in the middle are also going to turn brighter and brighter color. So in the morning, they will all be ready.
What was the whole setup like for all the living bacteria in the exhibition?
The setup time.
The setup, I mean, how long did the bacterial cellulose grow and how long?
Okay. Like Daniela mentioned before, I mean, the temporality and different timescales are a fundamental factor of the exhibition. Basically, every organism has its own timescale. No, cyanobacteria it reacts to a very specific photoperiod and normally relates to day and night alternation. The case of the Ecoli, for example, it's easier in a way, it's easier and faster. While in the case of the bacterial cellulose, we are talking about a growing time that is fairly long. So we are talking about 4 weeks of growing. And of course, also the time of drying, this is also something to take into account. So maybe 4 weeks plus another week or 2 for drying. And of course, I mean, this is an aspect that we have to deal with. No, we have to deal, especially if we have an environment that is interactive. No, what are our expectations as visitors, as humans? No, when we confront with these different timescales.
Maybe also to add, I mean, with this cyano and the interface you have here, you can see that the bacteria react immediately. So, for instance, there you can actually see the spikes whenever we turn on and off lights in real time. The biggest issue is actually how we can perceive it. That's why we always talk about different time zones, because they have their reaction immediate. But till we understand it, till we can see something, it takes time. So especially when we grow material like with cellulose, of course it takes time, it's the growing time. But if you can visualize the metabolism, we found some methods where it's actually fairly fast for us to understand what's going on. Just wanted to add this.
Thank you. And a follow-up question by Helga Eichner is if this duration is typical for your project.
Duration?
Bacterial life. Yeah, the time to grow and this dialogue situation is if that's a typical time frame you have.
Typical time frame
to set up and what are your experiences from the process?
For the interaction?
Or for the growth part and dialogue, I think both are addressed.
I mean, I think one of the biggest things that we have to face is the moment of disappointment. We humans are very used. We do an action and we want to see an immediate reaction. And also this training that till you can see the reaction of another subject might take time. And I think this was something we were continually facing in the project and also something we had to learn. So an interaction is suddenly shifts and it becomes something completely different where you have to kind of have a waiting period. So with the growing, I mean, even with the exhibition setup, we were starting quite some time ago because we had to start to grow the bacteria directly here in the exhibition space to a moment where we can start to interact. So also this growing period that we were setting up almost 3 weeks ago. We were setting up all the cyanobacteria directly here. And so this kind of time period becomes something completely different. You kind of have to prepare the environment. And it's a continuous discussion at the scale. So I say we were fighting it and supporting it, the waiting of a reaction continuously throughout the whole project.
Yeah, thank you for your questions. Now I would hand over to Barbara for general continuation.
Well, I'd like to thank everybody who's here for participating in the making of it. It was a long making. The whole project duration is 3 years. And we will, as mentioned before, conclude it with a a large publication. But before that, you have the opportunity to come here until the 11th of March, between Monday and Saturday, between 11 and 6 p.m., 11 a.m. and 6 p.m. to look at the exhibition and to also wear what Daniela is wearing, that interface, and establish your own non-verbal communication within all these living species here in Europe. So please sign up. You can go to our website, www.CoCorporeality.net, and there you'll find a doodle link, and you can book your own spot, and we will guide you through the exhibition. Thank you very much for being here, and I hope to see you in the exhibition.